ओके बेटा, सुक्रोज जो है दैट इज द नेक्स्ट disaccharide, and sucrose is also known as cane sugar because it's present in the sugar cane. And here you can see it's made up of two monosaccharides. And which monosaccharides are present in sucrose? One is the glucose and other is the fructose. यहाँ पर आप देखिए, मैंने आपको बताया था बेटा मोनोसेक्राइड्स कौन कौन से हैं. हमने देखा था देयर इज ग्लूकोज, फ्रुक्टोज एंड ग्लैक्टोज. ठीक है, तो दीज बोथ आर कंबाइनिंग विद वन एंड अदर, combining with one another, and they are forming disaccharides. Sucrose is made up of glucose and fructose. Both have the same formula. So sucrose also has the same formula as that of the maltose. They both combine by a bond between them along with the removal of one water molecule. And let's see the other example, that is the lactose. Lactose is also known as the milk sugar because it is present in milk. It's a part of the milk and it is made up of two monosaccharides. Which monosaccharides are combining and forming the lactose? These are the glucose. Glucose is combining with the galactose, right? These both are combining together and they are forming the lactose. It also has the same formula as that of the sucrose and maltose. There is a bond formed between them which link galactose with the glucose along with the removal of one water molecule. So we have seen here that they all are formed by a same process, that is by the removal of the water molecule. But you have to remember that lactose is made up of galactose and glucose, and maltose is made and sucrose is made up of fructose and glucose, and maltose is made up of ग्लूकोज एंड ग्लूकोज. ठीक है, तो आपने बेटा याद रखना है ये जो डाइसेक्राइड्स हैं, विच मोनोसेक्राइड्स आर प्रेजेंट इनटू देम. माल्टोज इज मेड अप ऑफ ग्लूकोज एंड ग्लूकोज, तो ये हमने याद रखना है. अच्छा, अब बेटा जो इनकी फॉर्मेशन है, ओके दिस इज ओके, they are formed, the process which is actually the process in which two smaller molecules combine together and they form one larger molecule along with the removal of one water molecule, that is known as the condensation process. तो हम formation of disaccharides को and even the formation of polysaccharides को condensation भी बोलते हैं because smaller molecules have combined together. Like you can see, it's the chemical reaction where two simple molecules join together to form a larger molecule with the removal of one water molecule, right? And here in this example, we have seen that these two smaller molecules are combining together along with the removal of one water molecule and they are forming a little one larger molecule. तो इनकी formation की process को हम condensation भी बोलते हैं. ठीक है. अच्छा, now reverse happens, that's the hydrolysis. Hydrolysis is the breakdown of the complex molecules in the presence of water. ठीक है. अब अगर हम इसी प्रोसेस को रिवर्स कर दें, ठीक है, इस प्रोसेस को अगर हम रिवर्स करते हैं, if we take lactose and add water into it and an enzyme into it, that will give us then galactose प्लस ग्लूकोज. तो हमारे पास दोबारा यही चीजें बन जाएंगी जिनसे हमने लैक्टोज बनाया था. इनके रिवर्स प्रोसेस को हम हाइड्रोलिसिस बोलते हैं. दिस इज नोन एज द हाइड्रोलिसिस. यू कैन सी हियर, इसमें क्या है, हमारे पास सुक्रोज है. सुक्रोज इज कंबाइनिंग विद वाटर. सुक्रोज वाज मेड अप ऑफ फ्रुक्टोज एंड ग्लूकोज, right? So sucrose will be broken down into glucose and fructose जिसका ये बना हुआ था. ठीक है, and we have studied enzymes and there we studied that enzymes are having the name as that of the substrate. Only ase, suffix ase is added at its end. So here you can say sucrose is broken down by an enzyme which is the sucrase. इसी तरह जो maltose होगा, that will be broken down by an enzyme which is the maltase, and lactose will be broken down an enzyme which is the lactase. ठीक है, ये रिवर्स प्रोसेस भी होता है, फॉरवर्ड में भी होता है. अच्छा, जो नेक्स्ट है बेटा, that is the polysaccharides. Polysaccharides are made up of more monosaccharides joining together. आप यहाँ पर देखें, ये बेटा हमारे पास स्टार्च है. ठीक है, and starch is having these smaller circles are actually representing the Smaller circles are representing the monosaccharides, और यहाँ पर आप देखें कि बेटा many monosaccharides are combining together and these monosaccharides are forming the larger और complex polysaccharides जिनको हम बोलते हैं complex compounds जिनको हम बोलते हैं polysaccharides. अच्छा, ये भी बेटा ऐसे ही होगा, when two will combine together one water molecule will be removed, three will combine another water molecule will be removed. इसी तरह ये combine करते हैं by the removal of water molecule. So it means कि यहाँ पर भी क्या हो रही है, condensation हो रही है. जब ज्यादा molecules combine करते हैं by the removal of water molecules, तो इस कंडेंसेशन प्रोसेस को हम बोलते हैं दैट इज द पॉलीमराइजेशन. पॉली मीन्स मेनी, ठीक है. पॉलीमर्स हैव जॉइंड, मेनी पॉलीमर्स हैव जॉइंड टुगेदर, सो इट इज नोन एज द पॉलीमराइजेशन प्रोसेस. ठीक है, पॉलीमर भी हम इसको बोलते हैं. Polysaccharides are formed by the polymerization by joining many monosaccharides. अब इसमें बेटा this is the structure of the glycogen. Glycogen is also the polysaccharide and this is the structure of the cellulose. Cellulose is also a polysaccharide. Now see the difference between starch, glycogen and cellulose. Starch जो है बेटा, starch is the storage form of carbohydrates in plants. If they have excess carbohydrates and that carbohydrate has to be stored into their body, so it will be stored in the form of the starch. Extra is stored always in the form of starch. It's also found in cereals, potatoes, grains, wheat, rice and some other sources. It is tested by, this is very important, you have to remember, this is tested by iodine. Iodine itself is of brown color and when we test it with the, when we add into starch, it turns blue black. So if the iodine turns into blue black, it means that the sample has starch into it, and if it remains brown, it means that the sample do not have starch into it. You can also test it at home because I think every one of us has iodine at our home. Iodine is of yellowish to brown color. You can take 1 drop of iodine and place it on potatoes, or you can cut a slice of potato and place 1 drop of this iodine onto the potato. You will see that its color will turn blue black because the potatoes have starch into it. It's made up of glucose. Okay, beta, you have to remember that all are made up of glucose. No fructose, no galactose, or no any other monosaccharide is present here. So all are all is having the glucose. It's also made up of glucose. Glycogen is also made up of glucose, and it is also, cellulose is also made up of glucose. Okay, if all are made up of glucose, then where is the difference lying? Difference is in the number of the glucose molecules and in the type of the glucose molecules, because there are the 2 types of the glucose which exist in nature. One is alpha and one is beta. So the difference in starch, glycogen and cellulose is they all are made up of glucose, but they have the different type of glucose. Some will have alpha and some will have beta, and they have different number of glucose. Like glycogen, it has 10 to 20 glucose molecules. Starch have 200 or more glucose molecules, and it has even more than 200. Cellulose can have more number of glucose molecules. Next is with the glycogen. Glycogen is also known as the animal starch because if the extra carbohydrates have to be stored into our body, that is always stored in the form of the carbohydrates. ठीक है, हमारी बॉडी में एक्स्ट्रा कार्बोहाइड्रेट्स आर स्टोर्ड इन द फॉर्म ऑफ ग्लाइकोजन. हमारी बॉडी में स्टार्च नहीं होता, यू हैव टू रिमेम्बर, और इट इज फाउंड इन लिवर एंड मसल्स. एक्स्ट्रा ग्लाइकोजन इज ऑलवेज स्टोर्ड, एक्स्ट्रा ग्लूकोज इज ऑलवेज स्टोर्ड इन द फॉर्म ऑफ ग्लाइकोजन, और ये हमारी बॉडी में स्टोर होती है इन लिवर एंड इन मसल्स. Cellulose, we have studied this, it's the component of the cell wall, and these all are made up of glucose molecules. Okay, here you can also see. अच्छा, अगर बेटा रिवर्स प्रोसेस होना है, like I've told you that starch is made up of glucose, glycogen is made up of glucose, cellulose is made up of glucose. So first they will be broken down into disaccharides and then they will be broken down into monosaccharides. Here we can see that this is the starch molecule, and here you can see that there are many glucose molecules combined together. First they are broken down into disaccharides. Two molecules में break down होते हैं. They are broken down into maltose because they are made up of glucose. So maltose is also made up of glucose and glucose. So they all will form maltose, 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 maltose. Then those maltose are broken down into glucose. In the end, we will get the simplest saccharide from which they were made up of. And all these reactions are carried out by the enzymes. Yes, beta.
Excuse me.
Yes, beta. Yes.
Miss, these all are breakdown by enzymes.
Yes, all are carried out by the enzymes. ठीक है beta, when they are synthesizing, then also enzymes are required, and when they are broken down, then also enzymes are required. ठीक है, automatically if you are going to place two monosaccharides together, they will not combine and form the disaccharide. Some enzymes and some conditions are required to combine them together. Okay, beta, this is just one last. Then we have to, okay, we are also left with the test. Okay, starch and glycogen, which are the complex polysaccharides, they are the good storage substances. Okay, there are some of the reasons why they are good storage substances. One is because they are insoluble in water. Okay, they are insoluble in water, so that's why they can be easily stored. If they would have been soluble into water, then they would have dissolved and they would have been transported to other parts. Right? This is, they are insoluble, so that's why they are the good storage substances. They are larger, unable to diffuse through the cell membrane. Like if they are going to be stored into any part of the cell like liver or muscle, they are indiffusible. They cannot pass through the membrane of the liver cells or muscle cells, so they will remain into that particular part. Then next, they can be easily hydrolyzed. Hydrolyzed means that they can easily broken down into disaccharides and monosaccharides. This process is also known as the hydrolysis, जिसको हम digestion भी बोलते हैं. It's the hydrolysis. Larger है, whenever they are needed in the body, they will be converted into disaccharides and then they will be converted into monosaccharides. Means they can be converted into simplest. So that's why they are the good storage components. Then they have the compact shape, so they will occupy the small space. Like here I have drawn. They are the larger molecules, but they can form a compact or smaller structure which can fit into any smaller size. Beta, next, yes, this is important. अच्छा, इसमें जो है बेटा, how you are going to test the carbohydrates. हमारे पास carbohydrates में we have studied there are monosaccharides, disaccharides and polysaccharides. Just give me beta 2 minutes more. Okay, and अच्छा, these are tested by the BENEDICT test. Not all sugars are tested by BENEDICT test. BENEDICT test is for the reducing sugars. ठीक है, हमने देखा है कि हमारे पास सारे sugars हैं. BENEDICT test is for okay, BENEDICT test is for the reducing sugars, and reducing sugars are those which cause reduction or they give hydrogen. Simple. ठीक है, आपने देखना है कि carbohydrates which can give hydrogen, they are known as the reducing sugars, and those reducing sugars will give you the BENEDICT test. Now, what are the reducing sugars amongst all the carbohydrates we have studied? This is not starch. Let me correct it. Okay, beta. Achha, how we are going to see that what are the reducing sugars? Your sucrose, the sucrose which is the disaccharide, sucrose and all polysaccharides are the non-reducing sugars. इसके अलावा जितने भी हम देखेंगे all are the reducing sugars. ठीक है, sucrose and all polysaccharides मतलब glycogen, cellulose and glycogen, cellulose and starch. right? They are the polysaccharides, they are non-reducing sugars. उसके अलावा sucrose, which was the disaccharide, that is also the non-reducing sugars. So they will not give you this Benedict test. इसके अलावा बाकी जितने भी हैं, all are the reducing sugars. They will give you this test, give you means like glucose, galactose, fructose, maltose, ठीक है, जो हमने पढ़े हैं, they all will give you this test. How this test is performed? We add Benedict. Benedict is a solution, right? We add that Benedict solution into the sample, and if our sample has the reducing sugars, so we add this into the sample and then we heat. Okay, and if our sample contains the reducing sugars, then this Benedict color changes from blue to red. Benedict itself is of blue color, and if our sample contains the reducing sugars, reducing मतलब उसके अंदर glucose हो, maltose हो, galactose हो, lactose हो, so it will turn from blue to red. This will give you blue red color or red precipitates. It means that our sample will have the reducing sugar. Otherwise हमारे sample में reducing sugar नहीं होंगे. ठीक है, इट कैन हैव सम अदर कार्बोहाइड्रेट, सुक्रोज हो सकता है उसके अंदर, ग्लाइकोजन हो सकता है, स्टार्च हो सकता है और सेलुलोज हो सकता है, बट इट विल नॉट हैव द रिड्यूसिंग शुगर्स. ठीक है, इससे हम टेस्ट करते हैं. और आयोडीन जो है, आयोडीन इज टेस्टेड बाय द सॉरी स्टार्च जो है दैट इज टेस्टेड बाय द आयोडीन.